The overall goal of this assay is to quantify the capacity for monocytes from human clinical samples to undergo transendothelial migration and foam cell formation.
This method can be used to answer key questions in the cardiovascular field, such as the atherogenic potential of cells from individuals who are at risk of acute coronary artery disease. The major advantage of this technique is that clinical samples can be measured both from fresh whole blood or stored patient cohorts. Furthermore, foam cell formation can be quantified by both microscopy and flow cytometry.
First, prepare the polymerized collagen gels from a fresh collagen suspension. To a 5 ml polystyrene tube, add sodium hydroxide, then 10 times M199, then acetic acid, and finally add fibrous collagen. Mix this well by pipetting. Next, aliquot 50 microliters into the internal wells of a sterile flat-bottomed 96-well tissue culture plate. In the outer edges of wells, load 200 microliters of 1 times Dolbeco PBS to prevent desiccation. After 2 hours of incubation at 37 degrees Celsius, the collagen will polymerize. Then, overlay the gels with 150 microliters of 1 times supplemented M199 and incubate them until needed for use 5 days later. To expand stored human umbilical vein endothelial cells,